everyone, and thanks for listening to my presentation today. My name is Jean-Pierre. I'm a postdoc at McGill University in Montreal, Canada. And today I'm going to talk to you about the results of some of the projects I've been doing over the past years working on muskox in the northeast Greenland. So the title is Individual and Population Response to Climate Change. So I have to acknowledge my co-authors, so it's mostly the group of muskox ecologists working at Aarhus University, and especially Gonzalo, who's been helping me kind of troubleshoot some of the DEB modeling issues that I've come up with throughout this time. And for those of you who don't know me, I put a little picture of myself in the upper right corner, so if you want to put a face to the name. So we all know that climate change is affecting the world, so we know that increasing temperatures or temperatures are increasing globally, but what's important for this talk today is that the Arctic is particularly sensitive to this issue. So it's warming at twice the global average, and this is really highlighted in the map here where some of the darkest changes in surface temperature are happening in the Arctic. On top of long-term climate change associated with human impact, we also have short-term dynamics occurring in the Arctic. So the Arctic is a seasonally extreme environment, so we can go, massive changes in things like temperature and snowfall throughout the year. Now, these kind of challenging conditions can lead to seasonal forage bottlenecks. And what I mean by this is that animals in the Arctic rely on a relatively short summer season, so often just 3 months of the year, to get all the energy they need to sustain themselves for the 9 months of the winter where there's very little energy in terms of food resources. Now, species are obviously impacted by this, but they have also been adapted through behavioral and physiological adaptations. Things like adjusting your body temperature and metabolic rate throughout the winter to conserve energy. But the big question, of course, is how will climate change interact with these harsh conditions that are already present in the Arctic to stress out the animals that live there? This is where I'll introduce the muskoxen as the key figure in this presentation. So the muskoxen is a key Arctic herbivore. It's the largest herbivore in the Arctic, and as such has an important role to play in that ecosystem. It's a capital breeder. This is really important in terms of energetics because it relies on the energy that it stores throughout the summer to fuel reproduction, which happens throughout the winter. So essentially calves are born at the end of the winter where there's still very little energy or very little resources, and these animals rely on their energy from the summer. Much like other ungulate populations, muskoxen population dynamics are driven by calf recruitment, which has been shown to be highly variable. And specifically, we're looking at muskoxen in northeast Greenland in an area called Zackenberg. Here, there's a monitoring research station that has long-term data going back decades on the flora and fauna in the area, including muskox census data for looking at population dynamics and calf recruitment. So with this data in hand, we're going to ask a few different questions. The first one here is going to be looking at the short-term implications of harsh winter conditions to ask, how do muskoxen adapt to this winter forage bottleneck? And the second one, we're going to be looking at population dynamics and more long-term climatic changes to ask what are the main drivers of muskox population dynamics? And then how will muskox populations respond to climate change? So jumping to the first part here, so looking at kind of short-term physiological adaptations to harsh winter conditions, we're interested in looking at seasonal heterothermy. So in a paper we published last year, we had core body temperature loggers present in wild muskoxen in northeast Greenland. And this is the data shown in the figure here, where essentially in yellow, I want you to focus on are these wild animals that were pregnant, and they kept their body temperature constant throughout the winter. And then in blue here, I'm showing the wild animal temperature profile in non-pregnant animals. And as you see, the temperature drops throughout the winter, where there's a difference of about 1 degree Celsius, and then the body temperature rises back up to the normal one in the summer. For the sake of this presentation, these animals that didn't change their body temperature we'll call homeotherms or normotherms, and the ones that did fluctuate their temperature seasonally will be called heterotherms. And really the big question is, what are the energy consequences of these metabolic strategies? So this is essentially why we developed this model. So this is essentially an adaptation of our previous DEB-IBM for muskoxen, where we used only the study period of interest here, which is the winter period from October to May. As an input, we use the average body temperature of the different individuals within these two groups. So the normotherms are in orange at the top here, you can see their body temperature is about stable throughout the winter. And then the heterotherms, we get this drop in body temperature and then an increase back at the end of the winter. Now these are essentially used to derive a temperature correction factor, which we apply to the metabolic rates in the model. The other input is snow depth, which we have as an average estimate over the entire study area. And you'll see, we have increases in snow throughout the winter. And we use this to convert it essentially to a food availability, so the functional response is affected by the amount of snow. These two inputs go into our DEB model, and what we look at is essentially things like body weight, reserves, metabolic rate, and fitness consequences like reproduction and survival. This is what the typical results look like, where we're going to compare these two physiological strategies, so the heterotherms in blue and the normotherms in orange, so the ones that didn't change their body temperature. And here we're going to assume that all these animals, so both groups of animals, are non-pregnant, so we can focus on the results of the body temperature impact and not reproduction. So we're eliminating that confounding factor. So really the take-home message here is that the difference in body weight and energy reserves increases throughout the winter, and at the end of the winter, what we have is a major difference between these two groups. So for body mass, this equates to about 18 kilos, and for energy reserves, it's about 100 megajoules. Now remember, this is important because there's still a lot of snow by the end of May, and these animals are going to rely on their energy stores essentially for the next round of reproduction. Now the results of this come from obviously a change in the metabolic rate. So this last panel is showing the field metabolic rate, or the FMR. And as you'll see, the normotherm animals have a higher metabolic cost as they didn't reduce their body temperature and therefore metabolic rates. So this cumulative energy savings is what's resulting in these fitness benefits. Now we also ran hypothetical scenarios for pregnant animals. So you'll recall that the pregnant animals did not drop their body temperature, but we ran these simulations anyways just to see what hypothetical energy savings could be if they did reduce their body temperature. Now this does happen in other animals, so it's not unheard of. And what I'm showing here is really focusing on the reproductive endpoints. So this top panel I'm showing the reproduction buffer, and in the bottom here I'm showing the growth curve for the fetus. So again, you'll notice that there becomes kind of a growing gap between the heterotherms and the normotherms. So essentially by reducing your body temperature and saving energy through reduced metabolism, we can enter the end of May when the calf is born with much higher energy in the reproductive buffer, essentially allowing for more energy transfer through the milk. And on the bottom figure here, looking at the growth curves, the solid lines are showing when the fetus temperature is constant. So even if the mother reduces her body temperature, the fetus does not. And of course, that has zero implications on the growth curve. But the dotted line is showing that this scenario where the heterotherm female reduces her body temperature and the fetus does the same. So essentially this reduced metabolic rate in the fetus leads to a reduction about 50% in birth weight at the beginning of May. Now of course this has important implications, as birth weight is an important predictor of lifetime fitness for these animals. So to quickly summarize before heading to the next part of my talk, I'll just say that the literature has shown that heterothermy is present in a lot of small and large mammals around the world. And essentially its usefulness is by limiting the amount of reserve usage, so limiting the amount of energy usage, which can increase survival and ensure future reproductive success. We've shown here that models can be a quite useful tool to dive into this question. So while we know, for instance, that these animals are changing their body temperature, we have a lot less data about fine-scale temporal resolution on things like body mass and reserve dynamics, and even less about implications for reproduction and survival. So models can give us this kind of information. For muskox specifically, we've shown that heterothermy can have marked benefits for these animals, especially in terms of body mass and reserves. And while I didn't show the results, we use a lot of different simulations to show how it could also affect reproduction and survival under harsh conditions. And lastly, we weren't able to show what the consequences of heterothermy are in terms of the negative impacts. And this is mostly because we actually don't know what these are. So they're likely to occur because deviating from homeothermy, which would be the optimal temperature for your body, must have some consequences. But at this stage, we simply don't know what they are, especially for these animals that only reduce their temperature by about 1 degree. So for the remainder of the talk, we'll go into the population dynamics over kind of the longer term period, so looking over the 20-year monitoring data set that we have. And this is what the model framework looks like to do this, where essentially as inputs we have environmental data in the form of daily average snow depth, as well as daily average NDVI, which is a measure of plant biomass in the summer. And we also include a forage quality metric, which reduces the quality of the plants throughout the summer growing season, as we know that this gets affected by the summer. We also include stochasticity in the model through inter-individual variability in the assimilation rate, as well as food availability. We also include density-dependent forage competition, where smaller animals are a little bit less competitive than their adults. And finally, there's some stochasticity in reproductive success in terms of estrus, abortion rates, and the amount of energy that's transferred to the calf from the mother. And these are dependent on the reserve density of the mother. And these are obviously used to fuel the metabolism model, so the DEB model, which then predicts things like the internal state of the individual, and as a whole, this predicts population dynamics over time. And this is what the monitoring data looks like. So we have on the top panel here, snow depth in gray and NDVI in green. And as you'll notice, the snow depth is quite variable over time, whereas NDVI doesn't really change. And this is in line with what we know is essentially that snow is a major driver of population dynamics in this population. In the second panel, I'm showing here the population composition in terms of adults, subadults, yearlings, and calves. And what you'll notice is the empirical data, the solid line, fits pretty well to our model predictions, except for this time period here where there seems to be pretty poor model predictions. And this is actually a time period that has been captured in previous studies that kind of deviates from a major climate impact, and it's likely things like infectious disease or mineral deficiencies that are driving the population during these years. And in this last panel, I'm showing the calf recruitment, which again, the model is predicting pretty well, except for these strange years where it's not doing such a good job. So now that we've shown that the model can predict the population dynamics pretty decently, we'll move on to some of the snow simulations that we ran. And this again focusing on snow because it is the major driver of population dynamics. So what we did is we looked at 3 different scenarios. So changing in the snow profile at the beginning of the winter, changes in the snow depth in the middle of the winter, and then changes in snow at the end of the winter. And again, the different colors are representing different severities, so plus or minus 5, 10, 20, and 30% changes from a baseline, which is the black line in the middle. We also looked at different consecutive year disturbances, so the repetition of these different snow severities from 1 to 10 years in a row, and then going back to baseline thereafter. And this is what the typical results look like for something like population size, where we have a disturbance that lasts for 1 year, there's a population either increases or decreases and then returns back to normal, and then we can see what the impact is for 10 consecutive years. And really the main take-home message here is that changes in snow across all the different timings that we were interested in had significant impact on these population size over time. Digging a little bit deeper though, we can compare these different scenarios, and what we find is that population impacts differ with severity, so the amount of snow change, the frequency of consecutive disturbance years, and the timing of the snow. So for instance, here we're showing that with increasing or decreasing snow, we get increased impacts on the population growth rate. Increasing frequency, obviously more years in a row of bad winters are going to lead to increased population impacts. And then lastly, the timing are quite important. So early and mid-winter snow has a much larger impact on the population growth rate than the change at the end of the winter. We can translate this information to a recovery time, which is essentially looking at the number of years it takes the population size to return to its baseline value, so the pre-disturbance. Essentially what we show here is that while the result is kind of non-linear and a bit messy, even short disturbance years of 3 to 5 consecutive bad years in a row can lead to recovery times of up to 10 years and reaching up to 20 years for these harsh conditions. Now, I don't really have time to get into the details. What we show is what's driving these population impacts is really calf recruitment, and it's very scenario dependent. So this is showing kind of when there's more snow versus less snow, and it's comparing the reproductive, the number of females that are pregnant the ones that still have a calf in the middle of winter, so the abortion rate, and the number of calves that are surviving after they're born. And essentially what we see is the effects of more snow early in the winter leads to increased abortion rates. More snow in the middle of winter leads to reduced calf survival, so essentially through giving less energy because females are in poorer condition. And then impact in the late winter snow increases the abortion rates the following year. So to quickly summarize here, just wanted to say that we've shown that we could integrate environmental data in terms of snow depth and NDVI in our DEB-IBM for Muskoxen and predict population dynamics pretty well. Again, the fit is pretty good to empirical data, except for the years where energy is not likely to be the major driver. We've shown that the timing and the amount of snow has large impacts on the population growth, and this is mostly through calf recruitment. While I didn't get into it in this talk, we did publish it in our Global Change Biology paper, where we also included summer simulations, so changes in NDVI, and we concluded that changes in the winter have much bigger implications than changes in the summer. And lastly, even short-term and moderate impacts on things like harsh winter conditions can lead to long population impacts in terms of population growth rate or recovery times. So with that, that's the end of my talk. I'll just thanks everyone for listening and tuning in. And if you have any questions or you just want to collaborate, please feel free to email me. My email is on the screen here, and I'm also on Twitter. You can look at my information here. Thanks.